Hey guys, today we are going to show you a video of how to do a PCR reaction. So you have some lecture material where I've explained how PCR works, and you also have a video from YouTube that is kind of a cartoon on how that works, and some slides. So you have 3 different ways of understanding how PCR works. This is the 4th way to actually see it in action. Keep in mind that this is a PCR for Listeria, but it can be applied to pretty much any pathogen. PCR is used to bypass the cultural plating steps when we're doing detection assays. So you'll do your pre-enrichment, your enrichment, and then you can do PCR instead of plating on MOGs. That's one way you can do PCR. You'll learn a little bit later in the semester that we use PCR as a tool for many, many different things, including some of the sequencing things that we do. Now, in this case, Meg is already starting with DNA that's been isolated from the organism. That process is called DNA extraction, and it's usually done with a kit. In commercial kits where we actually apply this to a food sample that's been enriched, we frequently just do something called heat lysis, which you'll take a small amount of the enrichment broth, you'll heat it up to about 95 for 5 minutes, and that releases the DNA of the microorganisms so that they're available for amplification by PCR. And so with that, I'm going to pass this off to Meg and let her narrate how you actually set up a PCR reaction and what it looks like when it's done.
Hi, I'm Meg. I'm not one of your TAs, but I do PCR extraction a lot. Well, PCR extraction, DNA extraction, and PCR a lot, so I'm going to show you how it's done. So we need a couple of things to do PCR. The first thing we need is our actual sample. It's right here, labeled I for isolate. We also need our master mix. The master mix is a mix of different things that help the reaction go. Those are going to be our DNTPs, so pieces of the DNA. It's going to be some stuff like TAC, which is the enzyme that actually does the reaction. And then it's also going to be a couple different stabilizers. Beyond that, we also have our primers for forward and reverse. And we have some water, and that basically just makes up for any reaction volume. So if we don't have exactly 25 microliters, whatever isn't there, we'll add water with. So the first thing we want to do is we want to get a clean tube, which is this guy, and open it up. And by general rules, you don't add your isolate until last, because if you want to put it in there first, you might contaminate things. So the first thing we're going to do is add 10 microliters of our master mix, which is green. The green dye doesn't really do anything. It basically is there to help you see it, and it's also there so you know you've added it in there. So we're going to add it a little bit. So we're done with our master mix, so now we're going to add our primers. The primers are what actually kind of flank the gene, and that allows us to basically say, okay, this is the gene of interest that we want, and this is all we want to amplify, instead of doing the entire genome, because that could be a lot. And we're only going to add 2 microliters each of our forward and reverse. So. And sometimes they're a little hard to grab onto. It's just a little bit, almost looks like nothing. And the reason we don't have to do this in a hood is because our bacteria isn't alive. This is all just genomic material. So it doesn't really matter if we're keeping it safe from anything else, because the PCR will only amplify what we want it to amplify. So now we've got our primers done. The last thing we have to do is add water. So if we added 10 of these and 2 of these, you have a 25 volume overall. We're going to add 1 microliter of our isolate. So that means we're going to add about, it takes a while to turn these pipettes, I'm sure you guys know. There we go. About 10 microliters, 11 microliters actually, of our water. we'll grab that. And now we're ready to add our isolate. And we're gonna only add 1 microliter because if you look at the videos, PCR is an exponential reaction, which means that you don't need a lot to have a big result in the end. Now we take our isolate, take a little bit of our genomic DNA, put it right in this guy, and you're done. So now we're upstairs and we're actually going to take our PCR reaction, which we've already mixed, and run it on our PCR machine. So PCR in general is to amplify your DNA, and the steps that it goes through is first it denatures the DNA, it breaks all of those double-stranded bonds, so that way it frees it up so that it can be amplified by our enzyme. And then the next step is called annealing, and that's basically so our primers can get on there so it can amplify the right thing, our gene of interest. And then after that, it's extension, which basically means it just extends it. It's when the reaction takes place, and those all occur at 95 degrees for denaturation, about 50 to 60 for annealing, and then around 75 for extension. And then they basically just repeat that a number of times. Sometimes it's 25, sometimes it's 30. But after you do that, when we're ready, we would open this guy up. It's like a happy robot. We'd put our samples inside, and then after it was finished, which would take about maybe an hour or two, we'd be ready to go. All right, so now that we've finished our PCR, we've taken our PCR product out, and we have to run it on this gel. The reason we run it on the gel is, one, to see whether or not our extraction worked, because if we don't see anything there, then maybe the PCR didn't work, there's something wrong with it, and we need to try again. Or we could also run it to basically see what species this is. So for Listeria, there's Listeria monocytogenes, which is pathogenic and isn't allowed in ready-to-eat foods. But then there's also Listeria innocua, which is not pathogenic and isn't as much of a problem. So when we run these on our gel, we'll run a current through it, and that will actually separate the size and the bands of our DNA, and it will differentiate whether this is Listeria monocytogenes or Listeria innocua. So to do that, we take some of our PCR product, anywhere from 5 to 10 microliters, and we load it into our gel. We have these little wells in here, and we very slowly eject. And you don't press down to that final level because you don't want any of the DNA to bubble out. And you do that for the rest of your samples, and then you're ready to go. All right, so now that our gel is running, we have it plugged into our power source. We have to wait a little while, about like 50 minutes, in order to see any separation and to be able to tell what species our Listeria is. We have a picture on the Moodle site of a completed gel with Listeria monocytogenes and Listeria innocua that you can check out, and you'll see it's very clear. There's different bands in different places. Listeria monocytogenes is up here, L. innocua is down here, and it's really easy to tell. So after that, we're done.
So now you guys have seen a demonstration of PCR, which is nucleic acid amplification. And in this case, we are detecting our PCR product using gel electrophoresis. On the lecture part of this, you will actually have, there's an explanation on how gel electrophoresis works. This is one of a couple of ways that we can confirm our PCR product. Actually, what's used more often now is something called real-time PCR, which involves the inclusion of a probe, a DNA probe that's internal to the amplified region that has a fluorescent label on it. And binding of that probe to the amplified DNA usually results in a release of fluorescence. That fluorescence is read by the machine and processed in a manner in which we can determine sample positivity. And I'll explain, that is also explained in the video on PCR where I kind of go through the steps of a reaction. But keep in mind that this is only one way that in real life you would confirm PCR product. And in industry, for example, now people have almost entirely replaced gel electrophoresis with real-time PCR.